Welcome, everyone. I'm excited to kick off our presentation today with the topic, Conservation Challenge, Biodiversity Metrics Analysis. We'll be exploring how different regions with unique biodiversity traits can guide our conservation priorities. Throughout this session, we'll look at a phylogenetic tree that's essential for calculating key metrics like UniFrac, PE, PD, ED, and RAW-Q. Our goal is to better understand these metrics to help us make informed conservation decisions. Let's get started. This slide doesn't contain enough information. That's why we're moving to the next slide. Let's take a closer look at some key conservation insights related to diversity and dissimilarity measures. First, we have RAW-Q, which is a way to quantify phylogenetic diversity within a single community. It helps us understand how evolutionarily diverse that community is. Next is UniFrac, which measures phylogenetic dissimilarity between two communities, giving us a sense of how different they are in terms of their evolutionary history. Now, these features are evaluated at different levels. Alpha diversity looks at the diversity within a single community, giving us a snapshot of its internal complexity. Beta diversity, on the other hand, compares two communities to see how they differ from each other. When it comes to calculation, RAW-Q is computed for each community individually, providing a single value that summarizes its diversity. UniFrac, however, produces a distance matrix when comparing pairs of communities, showing the dissimilarities between all pairs. The outputs reflect these differences. RAW-Q gives us one value per community, while UniFrac provides a matrix of distances between communities. This allows us to make meaningful comparisons, whether it's looking at the average diversity across communities or understanding how distinct two communities are from each other. And at the core, these measures answer some fundamental questions. For example, how diverse is this community? Or how different are these two communities? These insights are crucial for informing conservation strategies, helping us identify which communities might need more protection or which are particularly unique. Let's take a closer look at some key conservation metrics and what they tell us about biodiversity. First up is UniFrac, which measures the phylogenetic distance between communities based purely on the presence or absence of taxa. Think of it as focusing solely on which species are there, without considering how many of each. It's particularly useful when your main interest is understanding how different communities compare in terms of species composition, regardless of abundance. Next is weighted UniFrac, which builds on that idea but incorporates how many individuals of each species are present. So, this metric gives a more nuanced view by considering both presence and abundance, making it ideal when abundance data is available and relevant to your analysis. Then we have RAW-Q, a bit different. This metric combines species richness and evenness into a single measure. It helps us understand the overall diversity and heterogeneity within a community, which is especially useful in ecological studies, where both the number of species and their distribution matter. Moving on to phylogenetic endemism, or PE. This metric highlights how unique the species in an area are within a phylogenetic context. It's particularly valuable when identifying biodiversity hotspots or conservation priorities that focus on species with limited distributions. Evolutionary distinctiveness, or ED, emphasizes the evolutionary history of a species. It quantifies how evolutionarily unique a species is based on its position in the phylogenetic tree. This is key when prioritizing species for conservation, especially those that represent a lot of evolutionary history. Finally, phylogenetic diversity, or PD, measures the total branch length of a phylogenetic tree for a community. Essentially, it reflects the cumulative evolutionary history represented by the species present. This metric is helpful when evaluating biodiversity from an evolutionary perspective and setting conservation priorities. All of these metrics connect back to our overarching goal, understanding and preserving biodiversity in a way that captures both species' identities and their evolutionary significance. Let's take a closer look at the phylogenetic tree structure. This tree is essentially a visual representation of evolutionary relationships among different species or genes. It helps us understand how these entities are related through common ancestors. The key points here are how the branches indicate divergence over time and how the branching patterns reveal the evolutionary pathways. The closer two branches are, the more recently they shared a common ancestor, which can tell us a lot about their similarities and differences. Connecting this to the overall theme, understanding the tree structure allows us to trace the evolutionary history, identify patterns of adaptation, and better comprehend the diversification of life. It's a foundational tool that helps us see the bigger picture of biological evolution. Let's take a closer look at the tree's structure represented here. Starting with the first branch, B1, which has a length of 4 and connects the root directly to SBA. This indicates that SBA is quite close to the root, making it a primary branch. Moving on to branch B2, with a length of 1, it connects the root to node X, which is an ancestor of several other branches. From node X, we see it connects to SBB1, SBB2, SBC1, and SBC2 through subsequent branches, highlighting how this node acts as a central point for these descendants. Branch B3, also 1 in length, extends from node X to node Y, which is an ancestor of SBB1 and SBB2. The subsequent branches, B4 and B5, are each 0.5 in length and connect node Y to SBB1 and SBB2 respectively, showing direct connections to these terminal nodes. Similarly, branch B6, with a length of 2, links node X to node Z, the ancestor of SBC1 and SBC2. Branches B7 and B8, each 1 in length, connect node Z to SBC1 and SBC2, respectively. Adding all these lengths together, the total tree length sums to 11. This overall structure illustrates how the branches and their connections form a hierarchical tree, with some nodes acting as central points connecting multiple descendants. Understanding this layout helps us grasp the relationships and distances between different nodes, which is fundamental for analyzing evolutionary paths or similarities within this tree. Let's take a closer look at phylogenetic diversity, or PD for short. PD essentially measures the total evolutionary history represented by a set of species in a particular region. We calculate it by summing the lengths of the branches in the minimal subtree that connects all the species in that region. Starting with region A, which only has SBA, the minimal subtree is just a single branch, branch B1, with a length of 4. So the PD for region A is simply 4. Now moving on to region B, which includes SBB1 and SBB2. The path connecting these species goes from the root through nodes X and Y, finally reaching the species. The branches involved are B2, B3, B4, and B5. Their lengths are 1, 1, 0.5, and 0.5, respectively. When we add them up, the total PD for region B is 3. Finally, for region C, which includes SBC1 and SBC2, the path goes from the root through node X and then to Z before reaching the species. The branches involved are B2, B6, B7, and B8, with lengths of 1, 2, 1, and 1. Summing those gives us a PD of 5. So what does this tell us? Essentially, regions with higher PD, like region C, represent a broader span of evolutionary history, capturing more diversity. This concept helps us understand not just how many species are present, but how evolutionarily distinct they are from each other, which is crucial for biodiversity conservation and understanding ecological relationships. Now, let's talk about evolutionary distinctiveness, or ED, which measures how much of an evolutionary history is unique to each species. The idea is that each species carries a fair share of its lineage's history, and ED helps us quantify that. The calculation involves summing the branch lengths along the path from the root of the tree to each species, but we divide each branch length by the number of descendants sharing that branch. This way, shared branches contribute less to the ED of individual species, reflecting their shared evolutionary history. Let's look at region A first. Species A1 has a branch length of 4, and since it's the only descendant, its ED is simply 4. That makes sense because it has unique evolutionary history. Moving to region B, we see species B1, B2, and B3. For B1, the branch length is 4, and since there's only one descendant, its ED is 4. For B2 and B3, the branches are shared. For example, B2 shares a branch of length 1 with B3 and C1, but because it has 4 descendants, we divide 1 by 4, giving 0.25. Similarly, B3 shares a branch of 1 with B2, but since it has 2 descendants, we divide 1 by 2, resulting in 0.5. B3 also has a unique branch of 0.5, so summing these gives an ED of 1.25. Now, looking at B2 and B3, both have an ED of 1.25, which reflects their shared and unique evolutionary history. B2 also shares branches with other species, which explains the shared contributions. In region C, species C1 and C2 each share branches of 0.25, 2, and 1. For the shared branch of 2, since it has 2 descendants, we divide 2 by 2, getting 1. The branches of length 1 are unique to each species, adding 1 to each ED. When we sum all these parts, each species in region C ends up with an ED of 2.25. Finally, if we sum up the EDs within each region, we get a total of 2.5 for region A, 2.5 for region B, and 4.5 for region C. This approach helps us understand which regions or species are more evolutionarily distinct, providing insights into their conservation value and evolutionary history. Let's take a closer look at phylogenetic endemism, or PE for short. This is a way to measure how unique and geographically restricted a set of evolutionary lineages are within a region. The formula for PE involves summing up the contribution of each branch in the phylogeny. Specifically, for each branch, we multiply its length by the proportion of its descendants that are found in the region, and then by the inverse of the range size, that is, how many regions the branch occurs in. The idea here is that longer branches represent more evolutionary history, and if a large proportion of their descendants are confined to a specific region, that region has a high level of endemism for those unique lineages. The inverse of the range emphasizes branches that are more restricted geographically, making them more significant for conservation. Looking at the table, you can see how each branch contributes to PE in different regions, based on their range and descendant proportions. For example, branch 1 has a length of 4 and is only in one region, so its contribution is quite high. Meanwhile, branches like B6, which are present in 2 regions, contribute less to the PE of any one region because their range is broader. Summing these contributions gives us a PE value for each region. In this example, region A has a PE of 2.25, while region B's PE is 4.25, and region C is even higher at 4.75. These numbers highlight which regions harbor the most evolutionarily unique and geographically restricted lineages. Overall, PE helps us identify priority areas for conservation, emphasizing regions that hold significant evolutionary history that's also geographically limited. Let's take a closer look at Rao's quadratic entropy, or Rao-Q for short. This metric helps us understand the expected phylogenetic distance between 2 randomly selected individuals from a community. Essentially, it tells us how evolutionarily diverse that community is. The formula for Rao-Q is a double sum. It multiplies the abundance proportion of one species by that of another species, Pj, and then by the phylogenetic distance between those 2 species, Dij. When we sum this across all pairs, we get a sense of the community's overall diversity. Now, looking at the examples, in region A, there's only one species, SBA, so there's no pairwise comparison to make. That means Rao-Q is 0 because there's no phylogenetic distance to consider. In region B, we have 2 species, SBB1 and SBB2. Their phylogenetic distance is calculated as the sum of 2 branch lengths, B4 and B5, which add up to 1. Since their abundances are equal at 0.5 each, the Rao-Q calculation becomes the sum of 2 identical terms, 0.5 times 0.5 times 1, repeated for each pair. Adding those up, we get 0.25 plus 0.25, totaling 0.5. Finally, in Region C, we also have two species, SBC1 and SBC2, but their phylogenetic distance is larger at 2, which is the sum of branch lengths B7 and B8. With equal abundances again at 0.5, the Rao-Q for this community is 0.5, calculated as 0.5 times 0.5 times 2, doubled for both pairs, giving us a total of 1. So these examples highlight how Rao-Q captures both the abundance distribution and the evolutionary relationships among species, giving us a more comprehensive picture of biodiversity across different regions. Let's take a closer look at uni-frac distances, which are really useful for understanding how different microbial communities are related in terms of their evolutionary history. Uni-frac quantifies what's called phylogenetic beta diversity, meaning it measures how dissimilar two communities are based on shared evolutionary branches. Think of it as comparing two communities by looking at their placement on a phylogenetic tree and seeing how much unique evolutionary history each one has. The key idea is to measure the fraction of branch lengths that are unique to either community when you combine their trees. There are two main types of uni-frac. First, unweighted uni-frac focuses only on whether species are present or absent, ignoring how many of each species there are. Its formula is the unique branch length in either community divided by the total branch length of the combined tree. Then we have weighted uni-frac, which takes species abundances into account. This provides a more nuanced picture, especially when some species are more dominant than others. Its calculation involves summing the branch length multiplied by the absolute difference in species proportions between the two communities, normalized by the total branch length. Overall, uni-frac helps us understand the evolutionary relationships shaping community differences, which ties into our broader goal of exploring microbial diversity and how communities evolve and differ across environments. Let's take a closer look at uni-frac distances, starting with the weighted uni-frac. This method is interesting because it doesn't just consider whether lineages are present or absent. It actually incorporates how abundant each species or lineage is within the communities we're comparing. The formula for weighted uni-frac involves summing over all branches in the phylogenetic tree. Specifically, we multiply each branch length by the absolute difference in the proportions of that branch's descendants found in each community, then divide by the total branch length of the tree. What does this tell us? Well, branches with high absolute differences in proportions, meaning a lineage is dominant in one community but rare or absent in the other, contribute more to the distance. For example, if a lineage is only in community A and not in B, that branch's contribution will be significant. This approach is different from unweighted uni-frac, which only considers whether descendants are present or not, so it's a binary check. Weighted uni-frac, on the other hand, measures how much of the evolutionary history is actually represented, taking into account the abundance data. Let's look at a full example comparing communities A and B. Imagine our tree has a total length of 11 units. We examine each branch, noting its length, the proportion of descendants in community A, and in community B, for instance, branch 1 has a length of 4, is fully present in a proportion 1, and absent in B, proportion 0. The absolute difference here is 1, so its contribution to the weighted uni-frac is 4. In contrast, some branches, like B6, B7, and B8, have no difference at all because their descendants are either in both communities or in neither, so they contribute 0. When you sum all these contributions, in this example, the total weighted difference is 6.5. Dividing by the total tree length of 11, we get a weighted uni-frac distance of approximately 0.591. This example highlights how incorporating abundance data gives us a nuanced view of community differences, linking evolutionary history with community structure. Let's take a closer look at uni-frac distances, which measure the phylogenetic beta diversity between different regions. Here, we're focusing on the unweighted uni-frac, which considers the fraction of unique branch length in the union of the subtrees. Starting with the comparison between A and B, we see that the unique branches are all the branches in either A or B, specifically B1, B2, B3, B4, and B5. When we sum their lengths, the total unique branch length comes out to 7. Since the total union subtree length is also 7, the uni-frac distance for a versus B is 7 divided by 7, which equals 1. This indicates complete dissimilarity based on unique branches. Next, comparing A and C, the unique branches are all branches in A or C, namely B1, B2, B6, B7, and B8. Summing their lengths gives us 9. The total union subtree length remains 9, so the uni-frac distance again is 9 divided by 9, which equals 1. So A and C are also entirely distinct in terms of their unique branches. Now let's look at B versus C here. The unique branches are B3, B4, B5, B6, B7, and B8. Note that B2 is shared between B and C, so it's not counted as unique in this comparison. Summing these unique branch lengths gives us 6. The total union subtree length for B and C is 7. Therefore, the uni-frac distance is 6 divided by 7, which is approximately 0.857. These calculations demonstrate how the unweighted uni-frac captures the phylogenetic differences between regions by quantifying the proportion of unique evolutionary history. This method allows us to compare communities or regions based on their phylogenetic makeup, which is a key aspect in understanding microbial diversity across different environments. Let's take a closer look at the uni-frac distances and what they tell us about the evolutionary relationships between these regions. Starting with the comparison between region A and regions B and C, we see a uni-frac value of 1. This indicates that these regions share no common evolutionary branches at all. In other words, region A contains only the ancient SBA lineage, represented by branch B1, while regions B and C only share the root branch, B2, but do not have any species-specific branches in common. This is quite significant, because it means that if we were to lose region A, we'd be losing 4 units of entirely unique evolutionary history. That's a substantial amount of diversity that we can't find anywhere else. It really highlights the importance of preserving region A to maintain that unique evolutionary legacy. Now, looking at the comparison between B and C, the uni-frac distance is 0.857, which tells us that about 85.7% of their combined evolutionary history is unique to one region or the other. The only branch they share is B2, the root branch that connects their ancestors, with a length of 1. Beyond that shared branch, each region has its own set of unique branches. Region B has branches B3, B4, and B5, totaling 2 units, while region C has branches B6, B7, and B8, totaling 4 units. This shows that each region has a significant amount of unique evolutionary history, reinforcing the idea that these regions are quite distinct in their evolutionary makeup. Overall, these results underscore the importance of conserving both regions, as they each harbor substantial and largely exclusive evolutionary information. Let's take a moment to review the overall results summarized on this slide. Here, we've got several key metrics across 3 different regions, region A, B, and C. Starting with PD, which stands for some measure of divergence, we see that region A has a score of 4. Region B is slightly lower at 3, and region C is higher at 5. This suggests that region C shows the greatest divergence among the regions, while region B has the least. Moving to PE, another important metric, region A is again at 4, but region B drops to 2.25, indicating less divergence there. Region C is at 4.25, slightly higher than region A, so it's somewhat similar but a bit more divergent. Looking at RAW-Q, which measures diversity or evenness, all regions are relatively close. Region A is at 0, B is at 0.5, and C at 1. This indicates that region A might be the most uniform, while region C has the highest diversity. The sum of ED, or some error or difference metric, shows 4 for region A, 2.5 for region B, and 4.5 for region C again. Region B appears to be the most consistent or least divergent in this regard. Finally, the UniFrac average, which measures community similarity, is 1 for Region A, and approximately 0.857 for Regions B and C. This suggests that Regions B and C are somewhat more similar to each other than to Region A, which stands out a bit more in this measure. Altogether, these metrics give us a comprehensive snapshot of the differences and similarities among the regions. They help us understand where the most variation exists and how the regions compare in diversity and divergence, an essential part of interpreting our overall findings and guiding future analysis. Let's start by looking at Region A, which we consider a priority due to its irreplaceable species. This region has the highest evolutionary distinctiveness, or ED, with a score of 4, and the highest phylogenetic endemism, PE, also at 4. This means it represents a unique evolutionary branch that we can't find anywhere else, making it incredibly valuable from a conservation perspective. However, one weakness is that it has low functional diversity, indicated by a ρQ of 0, which suggests limited variation in traits and ecosystem functions. The biggest risk here is that if SBA were to go extinct, we'd lose 4 units of phylogenetic diversity, PD, which would be an irreversible loss. Now, moving to Region C, this area is notable for its high functional diversity, which is reflected in its top scores across several metrics: PD at 5, PE at 4.25, ρQ at 1, and total ED at 4.5. These numbers tell us that Region C harbors a broad range of evolutionary history and trait diversity, making it a very strong candidate for conservation. The only caveat is that it's less irreplaceable compared to Region A because it shares its root branch with Region B, meaning some of its evolutionary uniqueness isn't entirely exclusive. Speaking of Region B, it's characterized by its endemic species, meaning species found only there. The strength of this region is that its endemism increases our local responsibility for its conservation. But it has the lowest diversity scores, PD at 3, PE at 2.25, and ρQ at 0.5, indicating less overall biodiversity compared to the other regions. When we consider all these factors, Region C emerges as the optimal choice for conservation. It maximizes phylogenetic diversity, functional diversity, and evolutionary distinctiveness. While Region A is irreplaceable, conserving Region C offers broader benefits, including the protection of 2 evolutionarily distinct species and high endemism. Plus, maintaining Region C helps preserve the evolutionary potential of the ecosystem because it contains a wider array of traits, which is critical for resilience. Finally, I want to clarify a methodological point. Weighted UniFrac and ρQ are both important tools in biodiversity analysis, but serve different purposes. Use UniFrac to compare between different communities or regions, what we call beta diversity. ρQ, on the other hand, helps us understand functional diversity within a single community, alpha diversity. Together, they give us a comprehensive picture of biodiversity, both in terms of species differences across regions and the diversity of traits within a region. Let's take a closer look at the UniFrac insight. What makes UniFrac so valuable is its ability to compare microbial communities based on their evolutionary relationships. Instead of just looking at which species are present or absent, UniFrac considers how closely related those species are. This gives us a more nuanced understanding of community similarities and differences. It's particularly useful when analyzing complex microbiomes because it captures the phylogenetic distances, providing deeper insights into how communities evolve and interact. This aligns perfectly with our overall goal of understanding microbial diversity on a more functional level. By integrating UniFrac into our analysis, we can better identify patterns and shifts in microbiomes that might be linked to health, environment, or other factors. So, in essence, UniFrac adds a layer of depth that makes our comparisons both more accurate and biologically meaningful. Let's take a closer look at the UniFrac insight here. The total tree length sums up to 2.1, which represents the combined branch lengths of all the species we're considering. This total helps us understand how much evolutionary history is shared or unique among these species across different regions. Notice how the species are categorized. Some are exclusive to the forest, some are exclusive to the savanna, and others are shared between both regions. For example, SBF1 and SBF2 are only found in the forest, while SBS1 and SBS2 are only in the savanna. SBC, on the other hand, is present in both regions. The abundance data shows us how prevalent each species is in each environment, which can influence how we interpret their ecological roles and evolutionary relationships. By analyzing these details, we can better understand the biodiversity differences between the forest and savanna, as well as the shared evolutionary history represented by the shared species. This insight is crucial for comparing microbial communities across different habitats and understanding their underlying evolutionary connections. Let's take a closer look at the unifrac insight presented here. On this slide, we have a breakdown of different branches, their lengths, and their descendant species, along with their presence in forest and savanna environments. Starting with branch B1, which is 0.3 in length and includes SBF1 and SBF2. The calculation shows that about 83% of this branch's descendants are found only in the forest, as indicated by the PF value of 0.83. Interestingly, none of these descendants are in the savanna, which is reflected in the PS value of 0. This suggests that this is a forest-only branch. Next, branch B2, with a length of 0.15, contains only SBF1. Here, roughly half, about 50% of its descendants are in the forest, while none are in the savanna. This indicates some degree of forest specificity, but not exclusive. Moving to branch B3, also 0.15 in length, which includes SBF2. Its descendants are about a third in the forest, with no presence in the savanna, indicating a more limited forest association. Branch B4 is a bit different. It's 0.2 in length and includes SBC, SBS1, and SBS2. The calculations show that approximately 17% of this branch is exclusive to the forest, but when we look at the savanna, about 100% of these descendants are found there, suggesting this is a shared branch between both environments. Now, considering branch B5, which is quite long at 0.5, containing only SBS1. Interestingly, none of its descendants are in the forest, but over half, about 57%, are in the savanna. This clearly points to a savanna-only branch. Looking at branch B6, with a length of 0.4, including SBC and SBS2, we see about 17% in the forest and 43% in the savanna. This indicates some shared presence, but with a stronger association to the savanna. Branches B7 and B8 are shorter, 0.2 each, including SBC and SBS2, respectively. Their presence is mostly in the savanna, with minimal or no presence in the forest, reinforcing the idea of environment-specific lineages. Overall, this analysis highlights how different branches are associated with either forest, savanna, or shared between the two. It illustrates the power of unifrac analysis in revealing these ecological and evolutionary patterns. Understanding these branch-specific insights helps us interpret how microbial lineages distribute across different habitats, which is central to our overall study. All right, let's take a closer look at the unifrac insight. What we're doing here is calculating the unweighted unifrac, which essentially measures how different two communities are based on their phylogenetic trees. The key idea is to look at the fraction of unique branch lengths in the union of the trees. For the forest sample, the branches that are exclusive to it are B1, B2, and B3, with lengths totaling 0.3, 0.15, and 0.15, adding up to 0.6. For the savanna, the exclusive branches are B5 and B8, with lengths of 0.5 and 0.2, summing to 0.7. Then we have shared branches B4, B6, and B7, which are present in both communities, with total length 0.2, 0.4, and 0.2, summing to 0.8. When we add all these up, the total length of the union tree is 0.6 plus 0.7 plus 0.8, which equals 2.1. To get the unweighted uni-frac score, we look at the total length of the unique branches, those exclusive to each community, which is 0.6 plus 0.7, totaling 1.3. Finally, we divide that by the total union length of 2.1, giving us approximately 0.619. This value tells us that about 61.9% of the total phylogenetic diversity in the combined community is unique to either forest or savanna, highlighting how distinct these communities are in terms of their branch compositions. This approach helps us quantify the differences in microbial communities in a way that's rooted in their evolutionary history. All right, let's take a closer look at the uni-frac insight data here. On this slide, we're breaking down the contribution of different branches to the overall uni-frac distance between two microbial communities. Each branch, labeled B1 through B8, has a specific length, which represents its evolutionary distance. The key calculation involves the difference in proportions, or p-values, between the two samples for each branch. For example, for branch B1, the difference is 0, 83 minus 0, 0, which equals 0.83. We then multiply that by the branch length, 0.3, to get its contribution to the overall distance, which is approximately 0.249. This process is repeated for each branch, with some differences being larger, like B4, where the difference is 0.83, and others smaller, like B7, with a difference of 0.12. The final sum of all these contributions gives us a total uni-frac distance of about 0.98, or 0.9805 to be precise. What this tells us is that certain branches contribute more heavily to the overall community difference, especially those with larger differences in proportional abundance and longer branch lengths. This kind of detailed breakdown helps us understand which parts of the phylogenetic tree are driving the differences between these microbial communities, providing valuable insight into microbial diversity and relatedness. Let's take a closer look at the uni-frac insight here. First, the weighted uni-frac value is approximately 0.9805, which, when divided by 2.1, gives us around 0.467. Now, compare that to the unweighted uni-frac, which is 0.619. Notice that the unweighted measure is higher than the weighted one. This tells us that when we incorporate abundance data, the dissimilarity between the communities actually decreases. Essentially, shared species, what we call SBC, play a buffering role, reducing the overall difference because these common species are present in both regions. Now, moving on to the high-impact branches. These are the parts of the phylogenetic tree that contribute most to the dissimilarity. In particular, branches B1 and B4 stand out. Branch B1 represents radiations unique to the forest, while B4 is interesting because it mostly reflects ancestors found in the savanna, with a probability of 1 in savanna versus only 0.17 in forest. These differences highlight how specific evolutionary lineages are distributed across the regions. This brings us to a conservation perspective. Protecting both regions isn't just about preserving current biodiversity, it's about safeguarding the evolutionary history embedded in these branches. For example, the unique radiations in the forest, branches B1 through B3, and those in the savanna, like B5 and B8, are irreplaceable. Additionally, some branches like B4, B6, and B7 represent shared evolutionary potential, meaning they are important for future diversification. Finally, it's crucial to recognize that losing the forest would mean erasing significant portions of this unique history. Specifically, the branch length units associated with B1 through B3, effectively losing entire chapters of evolutionary storylines. So, these insights underscore the importance of conserving both regions to maintain not just biodiversity, but the deep evolutionary heritage they contain. Let's take a closer look at the uni-frac insights. First, unweighted uni-frac is really useful when we care most about presence or absence of lineages, like determining whether different regions capture distinct evolutionary histories. It helps us see if the lineages are unique to specific areas without considering how abundant they are. On the other hand, weighted unifrac becomes crucial when abundance actually influences ecosystem function. For example, in our case with SBC, the dual region role highlights how the abundance of species can impact ecosystem processes. Now, what's really interesting here is that even though 62% of the branches are unique to just one region, meaning many lineages are region-specific, the shared species actually reduce the overall functional dissimilarity to about 46.7%. This suggests that despite many unique lineages, the shared species play a key role in maintaining functional similarity across regions. And that highlights the importance of SBC as a keystone lineage. It's not just about the number of shared species, but about their critical role in connecting and maintaining ecosystem functions across different areas.